All right, so we're doing the Unit 5 review. This was we talked about DNA, RNA, and protein synthesis. So for the very first thing that we covered was the structure of DNA, and we kind of reviewed this to begin with. Remember that the DNA stands for deoxyribonucleic acid. The only place that you can find DNA in a cell is the nucleus. When you have 3 nucleotides together, so a nucleotide is like a sugar, a phosphate, and a nitrogen base. When you have 3 of these together in DNA, we call it a triplet. You definitely need to know that. DNA is double-stranded. Anywhere where you have an A, you'll have a T. Anywhere you have a C, you'll have a G, and you can see that in the colors here next to this. The sugar in DNA is deoxyribose, and the whole purpose of DNA is to store genetic information. It's your genes, and your genes give you the information you need to make proteins or carry out protein synthesis. RNA, what we introduced after we discussed and reviewed DNA, is a little bit different. We call RNA ribonucleic acid. Because RNA is smaller, it can go anywhere within the cell. So we say like the nucleus and the cytoplasm can go in and out. When we have a group of 3 nucleotides on RNA, we call it a codon, specifically messenger RNA. We call that a codon. RNA is single-stranded. Everywhere that there's an A, its complement will be U, which is different than DNA. Everywhere there's a G, there will be a C, which is the same. The sugar in RNA is ribose. So it's the ribose sugar. And the whole purpose or the job of RNA is to copy the genetic material, the DNA, your genes, and use them to guide protein synthesis, which is the making of proteins. This takes us into what this whole unit was really about, the protein synthesis. We call that the central dogma of biology. I said the main goal of this entire unit is for you to understand what we use DNA for. Big picture, your DNA is made up of small segments called genes, and each gene is used to make a protein. But in order for it to go from DNA to protein, we have to go through transcription first, translation second. So DNA has to be copied into RNA, and then we have to translate that into a protein. So the first step 1 is transcription. Transcription has to occur in the nucleus. To transcribe something means to copy it. So transcription, we're copying DNA and we're turning it into messenger RNA. This takes place in the nucleus. When we make the DNA, or when we copy the DNA into messenger RNA, everywhere there's a T in the DNA, opposite of it will be U. Everywhere there's an A, opposite of it will be. That totally does not need to say. Anyways, the opposite of A should be U, and the opposite of C should be G. So every gene, remember, starts with TAC, and every codon sequence, the start codon, should A and the U here should be switched, so it should say AUG. DNA is transcribed into RNA. We know this is RNA because it has a U. We know this is DNA because it has a T. Let's see. And a group of 3 nucleotides in messenger RNA is called a codon. So transcription is copying DNA and turning it into RNA. Remember, we use this chart. This chart is called a codon chart because you use it to read codons, which are in the messenger RNA. So our very first codon should always be A, U, G. The very first letter is on this side, so A. Second letter across the top, U. Then you come to the area where A and U come together. You look over here for G. AUG codes for methionine. You do not have to memorize this. You will have this on the test to use. The second step of protein synthesis is called translation. Translation uses messenger RNA, which is in groups of 3 called codons, and transfer RNA, which has groups of 3 called anticodons, the transfer molecule, and the amino acid. During translation, big picture, we're taking messenger RNA and translating it into a protein. I also told you that polypeptide is short for is another word for protein. Okay, the building blocks of proteins are amino acids. So every time we add an amino acid, we make our protein just a little bit bigger. So messenger RNA is grouped in 3, is called codons. The transfer RNA will bring the amino acid to the ribosome, and that amino acid will be attached to our growing polypeptide. Groups of 3 on transfer RNA are called anticodons. After we make all the proteins, we talked about what happens when things go wrong, and we call those mutations. So mutations is just a scientific word for change. These mutations can occur in your genes, and they can occur in your chromosomes. The gene mutations, which is just a small segment of DNA, there are 3 types: substitution, deletion, and insertion. So here at the top was your messenger RNA sequence. And then we used the codon chart to see what CGC codes for. It was arginine. GUU codes for valine. UCC codes for serine. Then it's showing you, in this case, the G was substituted for an A. So where there should have been a G right here, an A was substituted. This is only one amino acid is changed as a result of this. So this substitution is considered a point mutation. Only one amino acid changed. So this is a small genetic change. The second one right here is a deletion. We deleted the G. As a result of deleting the G, everything else shifts down. Because everything else shifts down, this amino acid becomes a new one, this amino acid becomes a new one. So we no longer even have the same protein. This is a completely different protein. Insertion means we added. So here we added an A, and again, it shifted everything down to the right. We call this also a frame shift mutation. It's a big deal because every amino acid after this will be changed. So a substitution is a point mutation, only one amino acid changed. But deletions and insertions are frame shift mutations, where things, if it's deleted, it shifts down to the left. If it's inserted, it shifts to the right. The second type of mutation we talked about was a chromosomal mutation. And I explained that a gene mutation is just one of these lines here. So a gene is just a piece of a chromosome. So when we're comparing gene mutations to chromosome mutations, the gene mutation is pretty small versus a chromosome mutation, which would be pretty big. So a chromosome has a whole bunch of genes. So if a chromosome gets messed up, multiple things are affected. But if a gene is just messed up, it's only what that gene controls that's affected. And then I also wanted to remind you that a nucleotide is a monomer, or the smallest unit of DNA. When you put a bunch of nucleotides together, you form a gene. Your genes make up your DNA, and your DNA is then folded into chromosomes. Last topic we covered was meiosis, which is for the formation of gametes. And remember that gametes are sperm and egg. When we make gametes, we want every single sperm to be different. We want every single egg to be different, because this will increase our genetic variation, which, like we talked about in class, increases our chances of survival. Crossing over occurs during this process, during the formation of gametes. That increases genetic variation. At the end of this process, we have 4 genetically different daughter cells. So you can see that here at the bottom. All these cells are different, and each of those cells only has 23 chromosomes. Where we started off with 46, we'll end with 23. So whatever your chromosome number is, like for humans, it's 46. When you make gametes, we divide that number in half. So it's half of whatever. If our chromosome number was 12, our gametes would only have 6. If our chromosome number was 34, then we would have 17 as our haploid number, or our gametes would have 17 each. So whatever your somatic cell number is, your gametes are half. And then we talked about crossing over, and I just wanted to kind of give you an up close, because crossing over does occur during the gamete formation. It does increase genetic variation so that no 2 sperm or eggs are the same. This is why you and your brothers and sisters, even though you have the same parents, look different, because the chromosome will switch a little bit of genetic material. So here was you, here's your sperm or egg. Your sperm or egg are just slightly different from you. They look like you for the most part, but there's a tiny bit of them is different. Good luck, study for this test.